why most drug development programs fail before phase 1 and how to prevent it. A biotech company spent 4 years and millions of dollars developing a compound that showed genuine promise in vitro. 6 weeks before their IND submission, their CRO flagged in an assay that never have been validated for the metrics they were using. The molecule was real, but the data was not. Hello, I'm Raghuveer Singh Parmar, Scientific Affairs Director at Resolve Mass Laboratories, where we specialize in bioanalytical science that determines whether a drug program survives long enough to reach to a patient or not. Why bioanalysis matters early. Most programs don't fail because the science was wrong, they fail because the measurement was wrong. Bioanalysis is the infrastructure beneath every IND enabling study. It tells you whether the drug reached the target tissue, how long it remained active, and whether the dose you are planning will produce the exposure you need or not. Without reliable bioanalytical data, you are not managing risk, you are accumulating it. And every downstream decision like dose selection, safety margins, and narrative in your IND filing rests on the quality of what you measured in week 3 of your preclinical program. The confidence you carry into phase 1 is built assay by assay long before you write your first clinical protocol. Mistake number 1, weak bioanalytical planning. The most common failure pattern we see isn't technical incompetence, it's sequencing. Bioanalysis gets treated as a downstream task, something to sort out once the formulation is locked and tox studies are scheduled. That sequencing error has a predictable consequence. Assays get developed too late to be properly qualified. Sensitivity limits turn out to be insufficient for the actual concentration ranges you are working in. Sample preparation protocols rushed under time pressures introduce variability that compounds across every subsequent study. By the time the data quality problems becomes visible, you are already 6 months behind. What a less experienced team often misses is this. Assay sensitivity isn't just a technical specification, it's a strategic decision. If your assay can't reliably detect a drug at the tail end of the PK curve, you underestimate half-life. And that error propagates directly into your dosing rationale, your safety margins, and ultimately your clinical starting dose. Mistake number 2, poor pharmacokinetic data. Unreliable PK data is the quiet killer of the drug development programs. High intersubject variability in a preclinical study sounds like a statistics problems. It's not. It's a signal that something upstream, sample handling, matrix effects, calibration is introducing noise that the biology didn't create. When you can't accurately estimate drug exposure, dose selection becomes really a guesswork. And regulatory reviewers read PK data very carefully. An IND with a shaky PK narrative draws questions draw delays cost money that early stage companies do not have. We supported a program where PK profiles across study cohorts showed unusual scatter, not scientifically implausible, but inconsistent enough to raise concerns. The root cause was sample collection timing drift across participating sites, a procedural issue, not a pharmacological one, but by the time it was identified, 2 study arms required repetition and 6 months timeline gone. Mistake number 3 is weak biomarker measurement. Biomarkers are where precision medicine programs stand or fall. They are evidence that your drug is doing what you believe it's doing. Engaging the target, modulating the pathway, producing the biological response that justify your mechanism hypothesis. Poorly designed biomarker assays don't just generate bad data, they generate misleading data. A signal that looks like target engagement might be a sample instability artifact. A flat biomarker response might reflect an assay too insensitive to detect a real, clinically meaningful change. The compound advances, the question it was supposed to answer never gets resolved. Regulatory problems can kill programs. Regulatory expectations around bioanalytical validation are not ambiguous. The FDA expects documented, validated methods, not methods that work reliably on your hands, but methods that can be independently audited and defended in review. Incomplete documentation is one of the most common triggers for IND clinical holds. Not fraud, not scientific failure, just insufficient paper trails around decisions that seems obvious at the time. Compliance cannot be retrofitted. If your bioanalytical data was generated under conditions you can't fully reconstruct and document, you will find that out at precisely the worst possible moment. Technologies that improve success. The analytical tools available today, high-resolution mass spectrometry, ligand-binding programs, digital PCR for nucleic acid-based biomarkers, have genuinely changed what's achievable, but technology is only as good as the method development and validation wrapped around it. Platforms alone don't generate regulatory-grade data. The expertise to select the right approach for a specific molecule, specific matrix and endpoint, and then validate it to the required standard. This is where our drug programs succeed or stall. How experienced CROs reduce the risk? 3 things separate a genuinely experienced bioanalytical partner from a simply a capable laboratory. First, strategic engagement at the program level, well before assay execution begins. Second, anticipating regulatory expectations before the submission is written, not after the first review cycle begins. Third, institutional memory. Having seen what goes wrong with molecules like yours, in matrices like yours, under regulatory framework like the one ahead of you. Bioanalysis is not a vendor category, it's a fundamental strategic capability. Weak planning in the early months of a preclinical program compounds into timeline failures, regulatory delays, and in some cases program termination. The teams that reach phase 1 with confidence treated measurement quality as a first order concern from day 1. If you are sitting on an IND timeline that's tightened that you'd like, or methods that were developed faster than you're comfortable with, that conversation is worth having it. Resolve Mass Laboratories works with the programs at exactly this stage. Reach out to our scientists. The most expensive bioanalytical decisions is one you delay. Thank you so much for watching this session, and I hope it helps you, and see you in the next one. Until then, goodbye for now.